Welcome back, and we are now going to complete part 2 of chapter 4, cell structure. So when we left off, we had been talking a lot about the cytosol and cytoskeleton and those parts of the cell, and now we start moving in towards what we more often than not consider the actual organelles of the cell and the intracellular structures. So we start with what's known as the endomembrane system. And the endomembrane system consists of the nuclear envelope, the endoplasmic reticulum, both the rough ER and the smooth ER, the Golgi apparatus, and lysosomes. Also, peroxisomes are sometimes considered part of the endomembrane system, but they're really actually what we call a semi-autonomous organelle, and we'll get to those towards the end. But the endomembrane system itself is kind of this big network. And all of these different parts, they all are separate and they have separate functions and structure, but they all work together to carry out the larger processes of the cell. And one thing they do all have in common is they are all part of the protein synthesis pathway. One thing to remember about our cells is that proteins are what do all of the work. Yes, we have DNA, we have this genetic material that determines everything, but proteins are the real workers. They're the ones who actually make things happen. And proteins, in order to make things happen and make them happen correctly and in a timely manner, our cells have to produce them, and they have to synthesize them. They have to process these proteins. Sometimes the protein needs a sugar attached to it or a lipid molecule, something like that. So the endomembrane system is a big part of the protein synthesis and processing that goes on in the cell for cellular function. The plasma membrane itself, what we would call the cell membrane, is also considered part of the membrane system, but since it is on the outside of the cell, it's not really part of the endomembrane system. And the endomembrane system all of the different parts of it are oftentimes directly connected to each other or indirectly. With indirect connection, they are passing molecules and other compounds back and forth through vesicles. So we start with the nuclear envelope. The nuclear envelope is actually a double membrane, whereas we think of phospholipid bilayer in our cell membrane. The nuclear envelope is actually a double membrane, so there's 2 layers of phospholipids there. The outer membrane will be continuous with the membrane that forms the endoplasmic reticulum, in particular the rough ER, the rough endoplasmic reticulum. The inner membrane, also composed of phospholipids, really is just used structurally to help with protecting the genetic material in the chromosome. The inner membrane is lined with lamin proteins, and these lamin proteins form what is referred to as the nuclear lamina. And the nuclear lamina helps to keep the nucleus in this kind of 3-dimensional structure, and it also allows for some anchoring of some of the microtubules and actin filaments used for chromosomal territories. Embedded throughout the entire nuclear envelope are nuclear pores, and nuclear pores are very, very important structures. They have specialized proteins that surround the actual pore itself. They're not just holes, but they're proteins surrounding them that regulate the passage of mRNA out of the cell, as well as regulating passage of anything coming into the nucleus. So not only is our cell membrane semi-permeable, or what we call selectively permeable, but the nuclear envelope is also selectively permeable. And with good reason. Our genetic material is inside of this nucleus, so we need to make sure that it is very well protected. Now, all of the material that's found inside of the nucleus, of course, is not considered part of the endomembrane system. So here we have just kind of a bigger to smaller type of picture here. We have, of course, here's our nucleus in the cell itself, right? And we have our genetic material. That's all this wiggly kind of red stuff here. And we have our nucleolus. The nucleolus is a region within the actual nucleus itself. Here's a transmission electron micrograph, kind of showing you where we have the nucleolus, and there's chromatin, which is our genetic material wrapped around histone proteins. Here's the nuclear lamina just lining the inside here. All this squiggly stuff outside is actually endoplasmic reticulum. The nuclear envelope is just on the outside of that structural lamina, like they're showing you here. You can see here they're also showing you the two membranes. This is a scanning electron micrograph on the outside of the nucleus showing you these nuclear pores. So you can see these nuclear pores, there are just millions of them. The nucleus is covered with them. And if we get even smaller here, we get to the actual rendition here of the nuclear pore. And you can see that this pore complex here is going to highly regulate, sorry about that, it's going to highly regulate what can actually move in and out of that nucleus. And that's, again, really important because we need to really protect our DNA. The cell only gets one copy of that, so it needs to be protected. Now, the nucleus itself in its entirety is the largest organelle in the cell. It's usually visible upon staining with a light microscope. It contains a couple of different parts. We, of course, have our chromosomes. Now, a chromosome is our DNA, and the DNA is wrapped around these protein subunits called histones. So the DNA molecule itself is not the chromosome. It's actually the DNA wrapped around its histone. So structurally, a chromosome is made up of a combination of DNA and histone proteins referred to as chromatin. The nuclear matrix is composed of nuclear lamina, and an internal matrix is connected to it. This is used to create what are called chromosome territories. Our chromosomes don't overlap each other. So up here in this little picture here with all these different colors, here's a colored micrograph. This was done probably with Photoshop. They took an electron micrograph and used coloring on it. But over here, they've taken those colors and they've given each of the colors a number, and each number corresponds with, I did it again. Each number corresponds with one of the chromosomes. Now remember, we have two of each chromosome. So I want you to see here we have right here in this light blue is chromosome 9, and over here is chromosome 9, right? So we have two copies of each chromosome, and notice none of them overlap. We have no overlapping. Here's 17 in the dark green, and here's 17 in a red as well. So we have, here's the X chromosome, so this is a female cell. Right? So we have we have one X. I can't see a Y. Oh, we have a Y, so this is a male cell, excuse me. So here's our Y chromosome. The X and Y chromosomes are the sex chromosomes, and they're the only ones that don't match. All right, so then we also have in our nucleus, we have the nucleolus. The nucleolus is most easily seen in cells that are in a non-dividing state. If the cell's in a dividing state, then we're not really capable of seeing the nucleolus. And the only thing that's really important, well, it's not the only thing, but what is important about the nucleolus is that it's a very specific region within the nucleus in which ribosomes are made. And this is important because, remember, ribosomes come together to help translate mRNA, and mRNA is also made in the nucleus. So we need to make sure that ribosomes and mRNA leave the nucleus separately. We don't want the ribosome making a bunch of proteins inside of the nucleus that could possibly be harmful to other material inside of it. So the whole function of the nucleus, its first and foremost function is to protect our genetic material. It has to be protected from the outside environment. It's used to organize chromatin because our genes that determine who we are located on our chromatin, and we need that chromatin to be accessible, and our transcription factors, the proteins that start the transcription and kind of use our genetic material, have to be able to find what it is that they're supposed to be transcribing. And regulating the expression of our genetic material is also occurring in the nucleus. Ribosome assembly in the nucleolus, both the large and small subunit are formed. We also find the ribosomal RNA and RNA proteins in there. And it takes place in the nucleolus because it's so close to the genes that are required for encoding them. So nucleolus is where ribosomes are made. But those ribosomes, just so you know, those ribosomes will not become functional until they enter into the cytoplasm. Now, the endoplasmic reticulum is our next stop in the endomembrane system, and it is actually divided into two regions. So the endoplasmic reticulum is really quite large. It's incredibly large, not quite as big as the nucleus, but it kind of wraps itself around the nucleus. And it is formed out of these flat fluid-like tubules, and they're all kind of smashed up together. They're sometimes referred to as cisternae, and the ER membrane is actually continuous, if you remember, with that outer membrane layer of the nuclear membrane. It encloses a single compartment called the endoplasmic reticulum lumen. Lumen just means inside of something, the inside. Now, the two regions of the ER are the rough ER, referred to the RER, and the smooth ER, and referred to as the SER. Now, the rough ER is rough because that flat kind of tube-like membrane that's all smashed up together is covered in ribosomes. So these ribosomes leave the nucleus and they go out into the cytoplasm, but not all protein synthesis takes place in the cytoplasm. Most of it actually takes place in the membrane of the rough endoplasmic reticulum. So a ribosome and mRNA will come together, and after they come together, the ribosome will embed itself into the endoplasmic reticulum membrane. So the membrane itself looks kind of rough. It has all these little studs or dots all over it. All of those dots are ribosomes. Now, once a ribosome embeds into the ER membrane, it will carry out its protein synthesis, and when it's done, it will actually leave the membrane. So although the rough ER is kind of covered in all of these ribosomes, they are not permanently attached. They kind of come and go as needed. The rough ER is responsible for initiating protein synthesis, not all proteins, but most of them. It also takes part in the processing and sorting of proteins. We make hundreds of thousands of different proteins in our cells, and all of those proteins need to be processed, and they all need to be determined where they're supposed to be going in order to carry out their function. The rough ER is responsible for preparing new proteins for the membrane to carry them into the membrane. So any of our proteins that are what we call membrane embedded, this would be for both organelles and our cell membrane. And most importantly, the rough ER begins the process of what's called glycosylation. In glycosylation, sugar molecules will be attached to lipids or to proteins. So glycosylation is a very important process because these sugar molecules, specific sequences of sugar molecules, are often used by our cells for recognition to each other and to our immune system. From there, we move further away from the nucleus, still part of the endoplasmic reticulum, but we no longer have ribosomes embedding, and proteins are moving through the lumen of the endoplasmic reticulum from the rough ER to the smooth ER. And the smooth ER, there's no ribosomes, and a primary function of the smooth ER is actually detoxification. You see this a lot in liver cells. We produce glycogen in the smooth ER, which is carbohydrate metabolism. We use the smooth ER in heart cells and cardiac cells to maintain a calcium balance for our heart to beat and some of the electrical signals that our heart uses. And then, of course, if we're going to create or modify any kind of lipid, that's going to occur in the smooth ER as well. So the smooth ER is smaller than the rough ER, but as far as size goes, but it is really very important in some of the different functions of our cells. For example, in detoxification of liver cells, if you've ever heard of the term cirrhosis of the liver, that just simply means inflammation of the liver. And what's happening in cirrhosis of the liver is it's very common among lifelong alcoholics. And people who drink lots of alcohol for a long period of time, their liver starts to compensate for this constant bombardment of alcohol. Alcohol detoxification takes place in the liver. So all of this alcohol keeps coming in, so the liver cells say, well, I gotta keep cleaning up all of this, so I'm just going to make more endoplasmic reticulum. And the size of the smooth endoplasmic reticulum starts to overcome the cell. The cell begins to keep on creating more and more smooth ER. It has less ER. The cell gets very crowded. And this starts to throw everything off balance. When it throws everything off balance, the cells now turn towards inflammatory signals. The immune system is responding. It's just not a very good thing. So these cells get really big. And if you ever get the chance to look at liver cells from a lifelong alcoholic or someone who has cirrhosis of the liver for some other reason, take a look at it, see if you can find the smooth ER. It will be very large in those cells compared to others. The rough endoplasmic reticulum and the smooth endoplasmic reticulum are continuous with each other. So here on the left we have kind of our cartoon drawing, and you can see these yellow dots here are the ribosomes all studded within. And this is, you can see in here, this is the lumen. Here's the nuclear envelope, and it's continuous here with the rough ER, and then the rough ER comes out here to the smooth ER. These little guys right here are vesicles. They are coming off of the smooth ER, probably going to the Golgi. Here's another picture kind of showing the same thing. Here's our nuclear envelope. We're going in the other direction here. Nuclear envelope, and here are the nuclear pores. And it's continuous. They're showing you the continuity here with this rough ER. And the rough ER is really big, but eventually as we get away from the nucleus, we start to lose the ribosomes, and we enter over here into the smooth ER. On the right here is a transmission electron micrograph, and they're showing you here the rough ER. Here's our nucleus up in the top left corner, up here. And, sorry about that. So we have our nucleus up here in the top left-hand corner. Then we have our rough endoplasmic reticulum, and you can see it's studded with all these little black dots. Those are all ribosomes. And then as we move away from the nucleus down here towards the left, we're starting to see parts of the smooth ER down in here. And so you can tell the rough ER is much larger than the smooth. So when you see the smooth ER much bigger, you're looking at cells that are having to carry out probably a lot of detox activity. The Golgi Apparatus. The Golgi Apparatus is the shipping center. Personally, it's my favorite organelle. It just does lots of different things. It has a really cool shape. I don't know. It just is interesting to me. It's sometimes referred to as the Golgi body, the Golgi complex, and sometimes we just call it the Golgi. And it's composed of, think of stacks of pita bread. So it's just like this big package of pita bread, and it has the outside membrane and then the lumen area inside. Now, they are continuous with each other, but when a protein enters into the Golgi, it enters in what's known as the cis phase, or here on this picture on the left, that's the top. So the cis phase is usually, if you look over here to the right, the cis phase is closest to the endoplasmic reticulum, somewhere in between the rough and the smooth ER. And this is because these two, the endoplasmic reticulum oftentimes is sending proteins that are in the midst of process but are not finished yet. And those proteins will be sent by vesicle to the Golgi apparatus. Now the Golgi receives everything at the cis end. Okay, so that's the receiving end. These vesicles are going to merge. They're going to fuse in the cis phase. The protein or compound that's being processed is going to be released into the lumen of this region right here. When the processing is done, a new vesicle will form on the other side and then fuse with the next in the stack and go through its process here in this lumen, and then come out a new vesicle, and so on and so on and so on until we get to the very last portion right here, and these vesicles are now leaving and they're going to some part of the cell. There's 6 different places that the Golgi will send things to. So this is referred to as a shipping and receiving center. The cis end is the receiving center, and the trans face or trans end is where everything gets sent out. Now, vesicles, these little bubble-like structures, are what actually transport all of these different proteins and lipids and other molecules between organelles and also between the different folds of the Golgi. So here they're showing you the places that these vesicles can go, right? So we can go to the cell membrane, it can be secretion, it can be a lysosome. If endocytosis occurs, things are brought in. They go into a lysosome, that sort of thing. Endoplasmic reticulum, nucleus, there we go. So there are 3 primary functions for the Golgi apparatus. The first is secretion. Anything that has to be secreted out of the cell will go through the Golgi and it will be packaged. Now through that processing that occurs in the Golgi, one of the things the Golgi will do will actually tag the vesicle itself that's going to be moved around. And that tag is like putting an address on an envelope. Think of those vesicles as envelopes. And all of the little protein structures or sugars or whatnot that the Golgi places on the actual outside of the package is putting an address on it so that the cell knows where that vesicle needs to go. Those that need to go outside of the cell are known as secretion vesicles or secretory vesicles. They're going to fuse with the plasma membrane because the vesicles are composed of a phospholipid bilayer, just like our cell membrane. So the membrane of the vesicle fuses with the membrane, with the cell membrane, and then just releases whatever it is it's got in its, whatever package it's carrying will be released outside of the cell. Proteins that are secreted are usually synthesized in the endoplasmic reticulum. They'll travel to the Golgi to be shipped and then to the plasma membrane for release. The Golgi also works to process enzymes and some proteins and lipids. It will continue the glycosylation process started in the endoplasmic reticulum. Glycosylation takes place in what's known as the medial Golgi, which is kind of in the center. Another process carried out by the Golgi is proteolysis. Proteolysis is the process of breaking down proteins into smaller pieces, such as when we produce insulin, we actually store it in our cells in a form called proinsulin. And proinsulin is a really long protein molecule. It's like a big long chain of insulin molecules. And the Golgi, when insulin needs to be released, the Golgi will use specialized enzymes to cut proinsulin into the smaller segments that we know as insulin. And last but not least, the Golgi's major function is protein sorting. It is going to tag these proteins and other molecules with an address somewhere that it needs to go. There are 6 different locations that the Golgi can send things to. It can either stay in the Golgi because it's a protein that's actually being used by it. It can be transported back to the endoplasmic reticulum. The ER has to use its own proteins as well. It can take it to the lysosome in animals. We're going to learn about lysosomes in a little bit. It can take things to the central vacuole, which is a structure found only in plants. It can take things to embed them in the plasma membrane, or it can send things out for secretion. So here we are, our 6 places the Golgi exports to. Stay, endoplasmic reticulum, oh, little typo there, lysosome, central vacuole, plasma membrane, and secretion. So here are lysosomes. Lysosomes are vesicles found only in animal cells. Now they contain a very special type of enzyme, and these are a type of enzyme known as acid hydrolases. The lysosome is a really fascinating organelle because it's very simple. It's just it looks just like a vesicle. They're very small. The cell has lots of them, and they're just kind of this round membrane ball with a bunch of acid hydrolases inside of them. Now, acid hydrolases are enzymes that perform hydrolysis reactions. If you remember from Unit 1, when we put macromolecules together, such as proteins or DNA molecules or sugar molecules, when any of our macromolecules are made, they're put together using a condensation reaction where we remove water, a hydrogen atom off of one amino acid and an OH off of the other amino acid. This is known as a condensation reaction. We're removing a water molecule. But if I want to break those proteins apart, I want to break them up again, then I'm going to use a hydrolysis reaction. I'm going to add that water molecule back so that I attach that hydrogen back to where it needs to go. I attach that OH where it needs to go. And now those 2 amino acids, they're not going to bond to each other. So I can break a peptide bond with hydrolysis. Now, the reason they're called acid hydrolases is because they will only work in an acidic environment. The pH of the environment has to get below 5. And these hydrolases will only perform when it's acidic. So if this lysosome gets damaged or broken, or something happens to the lysosome inside of the cell, and it leaks or it breaks open, the cytoplasm of the cell is a neutral pH of 7. So if these acid hydrolases get released into the cytosol is a much higher concentration than these hydrolases, and those enzymes will be neutralized. They will not be able to be activated. They will be what we call denatured, and they won't be able to function. Now that's very important because of the primary function of lysosomes. Lysosomes break things down. They break down old organelles. They look for proteins that are no longer functioning, and they will absorb or fuse these organelles or molecules inside of their lumen, releasing these acid hydrolases. And when they fuse with something to phagocytize it, or to what they call endo or autophagy, when they autophagocytize it, they will turn on proton pumps, which increases the proton concentration inside of the cell, causing the pH to drop. And when the pH gets below 5, those enzymes, those acid hydrolases, are activated, and they start breaking down whatever it is that the lysosome has merged with. Different acid hydrolases are used by these lysosomes to break down different compounds. So some break down proteins, some break down carbohydrates, others break down nucleic acids, and of course lipids. So this allows the lysosome to be multifunctional, to have lots of different functions, and it needs to have that in order to kind of keep the cell nice and tidy and clean. Now, autophagy is that process of breaking down worn out organelles, and our cells in general carry out a process called ubiquitinylation. And in ubiquitinylation, any proteins or organelles or anything that are no longer functioning properly are tagged by the cell with a protein called ubiquitin. And lysosomes recognize structures that have ubiquitin bound to them. And when they see that, that is like a tag saying, hey, this guy's done. He needs to be recycled. So in autophagy, they will break down the components of an organelle and break it down into its single molecular units, so the cell can recycle those molecules and use them to build new ones. And then finally, endocytosis often occurs, and this is where the lysosomes are used to actually break down substances from outside the cell. Our lysosomes are used in phagocytosis. So in phagocytosis, our white blood cells are eating bacteria. A white blood cell will surround a bacteria and engulf it and take it in and create a vesicle inside of the cell harboring this bacteria, and the lysosome will merge with it. It becomes what's referred to as a phagolysosome. And the lysosome will destroy the bacteria, so it can't cause any problems to the organ. And now I had this video for you to show you the Golgi, but it's not working in this PowerPoint, so I'll play something for you in a little bit. All right, then we have vacuoles. Vacuoles have lots of different functions. Vacuoles are found only in plant cells. We don't have these in our cells. There are forms of vacuoles. We can't say they're not in any animal cells because we consider protozoans to be animals, and we do see food vacuoles and other vacuoles found in some of those protozoans. So we don't want to say they're only found in plant cells, but we don't find them in most animal cells. The functions are varied, and it depends on the cell type and sometimes what environmental conditions the cell is in. Central vacuoles found in plant cells are used primarily for storage and support. The vacuole itself is a big storage balloon, you could say, and inside of that balloon is mostly water. And the central vacuole, when a plant is healthy and has all the water it needs, the central vacuole is nice and plump. And that vacuole will provide not only storage of water and nutrients and minerals, starch in particular, but it will, that nice plump balloon will also give the plant cells some stability. So think about when you water your plants. If you have plants at home and you don't water your plants and they have to use up all of the water that's in their central vacuole, the vacuole starts to shrink and the plant will start to wilt. When you provide water back to the plant, the central vacuole fills up. This creates what's known as turgor pressure. So it creates pressure inside of the cell, the plant cell, And plant cells have cell walls, so it kind of bulges out against that cell wall, and the plant now stands up nice and straight. So in plants, it's used primarily for storage and support. In protozoans here, in protists, it's used to expel water. So we have a food vacuole to take in food, and we have over here, these are vacuoles that are used to pump water out in case the paramecium, too much water comes across the cell. There are phagocytic vacuoles in protists. That's what that food vacuoles are. And it is believed that we have vacuoles in white blood cells. They're pretty much the same thing as our lysosomes. Now let's talk about the plasma membrane. The cell membrane itself is the boundary between everything inside of the cell and the extracellular environment. So it's very important that it has some strength and integrity, and that it's very careful about what it lets come across. It's composed of a phospholipid bilayer, and we learned in Unit 1 that a phospholipid is a glycerol molecule with a phosphate and 2 fatty acid chains. Now those 2 fatty acid chains and a phospholipid bilayer, the chains are facing each other, which causes the inside, the internal portion of the membrane, to be nonpolar, and the outside, either side of the membrane, intracellular or extracellular, to be polar. That makes a phospholipid in what we call an amphipathic molecule. It is polar in one region and nonpolar in the other. The plasma membrane is selectively permeable. Now we're going to spend a whole chapter on the plasma membrane, so I'm not going to go too into detail yet, but it is responsible for transporting things inside and outside of the cell, and that selective permeability allows it to be very picky about what it lets across. We don't just have diffusion or free movement of molecules back and forth across the membrane. That selective permeability, that primary characteristic, is due to the function of a phospholipid bilayer. In cell signaling, which is Chapter 9, we're going to spend some time on that. We have a big assignment on that. The plasma membrane plays a major role. It's where all of the receptors are found for our cells to recognize what's going on in the outside. These receptors can be made out of proteins, sugars, and cholesterols, oftentimes in multiple combinations. And cell signaling is used for environmental responses. It's also used so our cells can communicate with each other. We want to make sure that we have good reflexes and responses. Cells communicate with each other, and they also need to recognize each other. Both forms of cell communication and recognition and environmental responses create what's called a signaling cascade. Like I said, insulin and glucose receptors. So when glucose comes into the body, we know that we need to produce some insulin. And so the pancreas receives that glucose signal and starts pumping out insulin to assist with glucose metabolism. And finally, the plasma membrane contains lots of proteins in it for cell adhesion. Our cells stick to each other, but they do so in such a manner that they're not just jumbled up together in any old way. They have a very distinct organization. Think about it. Everybody's kidney is the same shape. Everybody's liver is the same shape. We all have the same shape of heart and brain. So that's what works properly for that organ. And so these cells must adhere to each other, to some kind of matrix, in order to create these structures. These cell adhesions are also used for cell recognition, and different types of adhesions. We're going to look at the different cell adhesions in 10.2. They are also used in sharing of nutrients and in cell signaling. So here we have our plasma membrane with a signal cascade. Here we have 2 cells are adhering to each other, right? The top is cell 1, the bottom is cell 2. So 2 proteins embedded on the membrane, on the outside of the membrane here, are sticking together. So these 2 cells can stick together and be attached. Then we have here a transport membrane that's bringing glucose into the cell. Right? Our cells need glucose for energy. And over here we have another protein. Now notice this protein is a signaling protein. It's a receptor. It is not carrying anything into the cell, but when some type of outside signal, when some kind of outside signal binds to it, it's going to cause it to change shape and release these proteins that interact with each other in it like a row of dominoes. And it's called a signaling cascade. And eventually one of these proteins is going to go to the nucleus and cause some kind of cellular response to occur. Now, the semi-autonomous organelles consist of the mitochondria, the chloroplasts, and the peroxisomes. We call them semi-autonomous. They can grow and divide, but they're not totally autonomous. Now, mitochondria are found in plant and animal cells. Peroxisomes are found in plant and animal cells, but chloroplasts are only found in in plant cells. Now, the mitochondria is most often referred to as the powerhouse or the power plant of the cell. It is where the majority of a cell's ATP is made. It is membrane bound, and it has two membranes, but it is not considered part of the endomembrane system. It consists of both an outer and inner membrane. It has an inner membrane space, which is just kind of the space in between the two membranes. And then inside of the most inner membrane, it's known as the mitochondrial matrix. The inner membrane is folded. It has lots and lots of folds in it, and these are referred to as cristae. And these folds are very important in order to increase the surface area for ATP production. That inner membrane is critical in ATP production. So the more inner membrane that a mitochondria has, the better able it is to produce ATP. And that is its primary function. It converts chemical energy into forms of energy that can be used by the cell. It can be used for synthesis, modification, and breakdown of cell molecules. Not a whole lot, but it can do some. It's responsible for housing the electron transport chain that we're going to learn about in the next unit. And it can generate heat in what are referred to as brown fat cells, and they are used in thermal reactions in the body. So here we have our mitochondria. You can see this inner membrane here has all of these infoldings, these cristae, just like the surface of our brain. And it serves the same purpose. We need to increase surface area. The more surface area our brain has, the smarter we are considered to be. So we have lots and lots of cristae in the brain. Well, the same thing is occurring here in the mitochondria. The more cristae we have, then the more surface area of cell membrane. Of inner membrane for housing the electron transport chain to make ATP. Chloroplasts. Chloroplasts are found only in plant cells, and they are used for photosynthesis. They are the primary structure where photosynthesis occurs in plant cells, and it is also composed of an inner and outer membrane. There's a third membrane inside the lumen, inside of the actual chloroplast itself, called thylakoids. And these thylakoids are these individual stacks. So we have these big membranes in here that's referred to as the thylakoid membrane. And the thylakoid membrane folds in on itself, more surface area, and it creates what are called a granum, a large, think of a stack of coins, looks like a stack of coins. So a granum is the whole stack. A thylakoid is each individual each individual coin that makes up the granum. Its primary function, again, photosynthesis, converting light energy into chemical energy. This is all going on inside of the chloroplast. And we'll spend more time on the chloroplast in the next unit. Here's just a close-up again of a granum that's a stack of these thylakoids. Here's another close-up picture of a chloroplast inside of the cytosol. Now that brings us to peroxisomes. Peroxisomes are very small organelles. Where they came from and why we have them, nobody really knows. Their primary function is breaking down molecules, especially lipids. They catalyze chemical reactions as well. They break down molecules by removing a hydrogen or adding an oxygen to it. And these types of reactions create a waste product known as hydrogen peroxide. Hydrogen peroxide is highly reactive inside of our cells. It is it is considered a free radical. So the peroxisome has to break down the hydrogen peroxide that it made, and it does so with an enzyme called catalase. And catalase breaks down hydrogen peroxide into water and oxygen gas. And if you ever poured hydrogen peroxide on a cut, the catalase in your red blood cells is what is causing majority of the fizzing that you see with hydrogen peroxide on a wound. So I'm going to just show you a quick video here. This is from med school, but I think you can get the gist of it. You get the general idea of how it works.
Peroxisomes. Peroxisomes are 0.5 micrometers in diameter and are surrounded by a membrane. The highest concentration of peroxisomes is in the liver and kidney. Proteins are directed to the peroxisomes by a unique signal sequence with the help of protein chaperones, peroxins. Functions: generation and degradation of hydrogen peroxide. It contains enzymes that can either produce H2O2 or break it down. Long-chain fatty acid metabolism, detoxification of harmful chemicals by superoxide dismutase, synthesis of plasminogens and platelet-activating factors. The marker is uric acid oxidase. Peroxisomal disorders. Zellweger syndrome. Zellweger syndrome is also known as cerebrohepatorenal syndrome and is the prototype of peroxisome biosynthesis disorders. It is characterized by craniofacial dysmorphism and profound neurologic abnormalities. Zellweger syndrome is the most common peroxisomal disorder to present in early infancy. Genetics. The peroxisome biogenesis disorders, including Zellweger syndrome, neonatal adrenoleukodystrophy, and infantile Refsum disease, are inherited in an autosomal recessive manner. They are caused by mutations in at least 12 different genes. The majority of infants with these conditions have mutations in either the PEX1 or PEX6 genes that encode ATPases needed to import protein from the cytosol into peroxisomes.
So I wanted to show you that video because I wanted you to see that here we have one of the absolute smallest organelles that has minimal function compared to most of the other organelles in the cell, yet if it doesn't function properly, it can cause some really serious problems very early on. Now, that's all of the organelles that we're going to go over, the cell structures and their functions. And the next video I'm going to show you here, it's just a few minutes. It is called The Inner Life of the Cell. It was created for Harvard medical students, and it is showing the cellular structure changes that occur in a cell that has to emigrate out of the blood vessel to a site of injury. It's basically a white blood cell that has to leave the bloodstream and go into tissue to fight off some kind of infection. But what I really want you to see in this video, it's just music, there's no narration. I just want you to watch it. It's in the Unit 2 folder if you want to watch it again without going through this whole lecture. I really want you to see if you can identify any of the organelles that are shown. You can see where the mRNA is coming out of the nucleus and ribosomes are attaching to the endoplasmic reticulum, and a protein's processing through the Golgi, and the cytoskeletal elements and motor proteins, and all of these different structures that we have talked about in this unit. All right, so yes, it was very dramatic, but hopefully you were able to see in there some of the different organelles that we have talked about in this chapter, kind of see them in action a little bit. If you want, you can go to YouTube and look up Inner Life of a Cell, and there are lots of different versions of this. There are some that are about 7 or 8 minutes long, and they are actually narrated, and they talk about all of the different things that are occurring in the cell if you're interested in that. All right, everyone, I hope you have a good night, and I will see you all in class.